Hi everyone, my name is Maredh Lopez and today is January 20th, 2025. I'm going to be talking about the prokaryote and eukaryote interaction that I've selected, which is the nitrogen-fixing bacteria, the Rhizobium species, and the leguminous plants. So in the first part of my diagram I drew, we have a little sun and a representation of photosynthesis and a little bit plants. We have something called flavonoids that leguminous plants send out in order to attract those Rhizobium bacteria into the root hairs. So then as soon as they get those flavonoids, which are chemoattractants, they're like, oh my God, hey, we need some nitrogen. So they kind of start invading the root hair, little root hairs. And you can see here in the diagram how that works. This is happening in all the root hairs and spreading and keep it going. So by the end of it, if you pull a plant out and you see a lot of nodules, that's little nodules where Rhizobium bacteria are currently housed in. So we have the cortical cells and the epidermis, root hair cells. So the Rhizobiums kind of come in through those epidermal root hair cells to start the infection. And these cells, specifically those epidermal cells, have receptors, and they're called nod factor receptors. So bacteria comes in, within the bacteria, something is happening, which is transcribing and translating proteins called NOT proteins, which produce NOT factors. These NOT factors kind of like say, hey, I'm here to help. So they kind of interact with that NOT factor receptor, and everything starts from there. So we start with an infection thread, it's a little dot that spreads like an octopus. It's an entry point. It's a tunnel-like structure, and it's just an entry point or the beginning of the journey of this little bacterias. Then we have over here the infection thread kind of gets bigger, and the rhizobia is released into all the cells. And then you can see how little by little the nodule kind of starts forming. So as soon as you can see here how the nodule forms and how there's ammonium in there. These bacteria are needed because plants can't really take NH2, which is atmospheric nitrogen, directly from the atmosphere. So they need these little guys in order to process that. To understand this procedure and this symbiotic relationship, I drew this diagram, which is really helpful. The bacteria, as soon as they go back into the nodule, they're called symbiosomes, and then they differentiate into bacteroids, which bacteroids are the bacteria that actually can do that process of nitrogen fixation, which I'm going to explain now. So they differentiate into bacteroids, so they're ready to go. But the bacteria, it's really, really, really sensitive to oxygen. So the plant cells have evolutionized in a way that they now create something called legmonoblin that actually regulates oxygen levels inside the symbiosomes, or the little protective bubble, in order for them to have an environment that makes them being able to produce that ammonium, a form that the plant can absorb. So they actually have an enzyme called nitrogenase enzyme. This enzyme gets ATP from the plant. So another thing that the plant is supplying in order to get that ammonium. Then it just transforms atmospheric nitrogen into a form, which is ammonium, a form that the plant can get back use. The plant gives protection, nutrients, a little house, carbon supplies, and even ATP for this bacteria to do that job. So it's a beautiful relationship, and it's mutualistic beneficial. As soon as this NH4, also called ammonium, comes in, it really helps the bacteria protein synthesis, nucleic acid formation, chlorophyll production, and enzyme activation, which is basically everything in a plant. So having these little guys really helps them kind of absorb the nature in a way that they can and flourish in life. And it not only helps them, but it helps the soil and it helps other plants. So it really helps all of us at the end of the day. But yeah, there's a slight difference right here. 1 to 5 micrometers, and then plant cells are 10 to 100 micrometers. It's an amazing symbiotic relationship. I think it's really interesting how the plant cell kind of created this nodule. So these nodules are actually made by the plant in order for those bacteria to have that anaerobic environment, kind of low in oxygen, for them to flourish and kind of give back nitrogen to her. So I just think it's really interesting. But this is what I did. Thank you so much for listening. I hope you guys enjoy it. And let me know if you have any questions. I hope you guys have a great rest of your day.